This conference will now be recorded.
Thank you very much for a kind introduction, and thank you for the invitation to this meeting. Very happy to be here. So as you've heard in the introduction, so I've spent quite a few years in preclinical safety, where most of the things that I'm going to present today stem from. So I was responsible for all the cell-based work in Roche's preclinical safety organization. So everything around toxicology and ADME. And so during those about 15 years, we developed a strong interest to improve the cell models that are being used, and we started to explore, refine, and apply what is nowadays known under microphysiological systems. And this is what I'm going to talk about. Maybe towards the end, also touch a little bit upon my new area where I work now within our clinical research organization as part of pharma research at Roche, where I also want to see how we can apply these new tools to support clinical decision making. So now move forward. So I'm sure you're all aware of a number of efforts that have been taking place over the last 1 to 2 decades in early pharma drug development to reduce cost and animal use and increase speed and effectiveness. And these are all areas that have been part of my career so far. So early safety was a big theme and still is. Lab automation and since a bit fewer years also in silico approaches, AI-powered designs. We will talk today about the cell models, organoids, and organs on chips. And this is then kind of the next thing where our group is focusing on how can we apply real world data, make use of genetics, but also other technologies to support a more personalized approaches to improve and optimize the benefit risk ratio at an individual patient level. So when we look at where does the field stand as of today, advanced human cell models. So I tend to use that term as to me this covers everything from 3D to organs on chips and microphysiological systems. I would say there's mainly 3 areas where now these tools have found an application. Be it in supporting disease modeling and target identification, given that you have access to be it the relevant cell types or patient-derived cells. So this is certainly an interesting area that will expand over the next years a lot. The first place where we applied these models is at the stage of candidate selection before going in vivo. How can we improve some of the organ toxicity assays that are typically being run at this stage? And also later on, if something happens, if there's signals coming from animal tests before moving into the clinic, can we address the mechanism and its relevance to a human setting? So this is certainly right and very encouraging, but we feel this is certainly still quite a bit far away from what our vision is, and this is a bold vision that we have. Kind of the end game here would really be to start with the patient, so make use of human tissue and data from patients, and by using that as part of a reverse translation approach, establish those human relevant in vitro system, validate them against the human data available. And once these systems have been proven to be valid and relevant, they can be used for prediction. So really, instead of adding on top of an already quite an extensive battery of different types of in silico, in vitro, and vivo tests, really think of how can we really change the early drug development paradigm. And in order to achieve that, certainly a lot more work needs to be done, be it in the field of just basic bioengineering and also the cell biology aspects to build these models. And I will touch on a number of examples where you see what currently works and where we still have gaps. For both the reverse translation as well as the predictive approach, data science tools are and will be required. AI is a buzzword that we hear a lot, and predictive algorithms that can deal with this type of data. Furthermore, access to patient tissue, be it primary from patients or stem cell derived, depending on the question in place, and the access to these cells is something I keep on saying is really important, and especially in the beginning when this field started to emerge, is something that I think has been a bit overlooked. A lot of focus, especially by academic groups, was on the engineering aspects. I think that was important to set the stage. But what is really a challenge and will be a challenge also in the future, especially if you're thinking of more complex settings, if you're dealing with more complex questions like with an immune component, the access to actually the right cells remains quite a challenge. And taking it a bit further, if we want to use also then patient data, real world data, access to this data is also something that needs to be defined and discussed, and can also be a challenge. I've been involved last year in a number of discussions in working groups where we realized actually that this is also not easy to get there. So this certainly requires a good collaboration and cooperation between different stakeholder groups, ranging from academia, clinical organizations. We've been talking to patient organizations, which I believe can really play an important role, for example, when you think of rare diseases and for example, gene therapies, which often are seen as an important element there. Data science experts, developers, pharma, and regulators. So after about 10 years of investment of, I think, most pharma companies in these advanced human cell models, where are we today? So I'm listing here a few bullet points that are taken from a review that I wrote together with former AstraZeneca, now Emulate colleague Lorna Evert, last year in Nature Drug Discovery. So I think we see a burst of different approaches, and some of them have more, some have less potential for application. And I think it's not fully clear yet which cells and which materials, which setups and which assays are the right ones. There's now more and more working groups and also parts of conference sessions that try to define these standards, but to me at least, I think this is not entirely clear yet. What is certainly more clear is what we would like to see if we want to validate those models. What are the endpoints? What is really important there, so that we have trust and confidence in these types of systems. There's lots of overviews and reviews published, and including from myself. But still, this is fine, but still the best and most convincing way for adoption, I think, is to demonstrate compelling evidence for a defined use case. I think we share a common vision, but in the end, we need to see more of those use cases to really learn more and see where do we need refinement, where do we need improvement. We also, some people sense a bit when I'm talking to developers in academia, but also from small startup companies, they really see and sense that broad industry adoption is slow, certainly too slow. in their view, and the reasons for that may include that this always means quite a significant upfront investment into sometimes quite complex machinery and other gear that is required. Sometimes the technical setting is quite challenging and complex and may not be usable in an industry setting. As some of these systems are still of exploratory nature and quite new, they lack sometimes the assays robustness that is needed. Sometimes after some investment into a new approach, we have to realize that there's a lack of a clear superiority. There may be a little bit of an improvement in terms of predictivity, but not good enough so that an investment would be justified. And certainly also along with that, also an unclear translational potential to the human situation, which is something that is not specific to MPS or advanced human cell models in general, but that certainly applies to all types of cell models that are being developed. So this is where we are today. Now, if we look at the situation at Roche, and that may apply also to a number of other pharma companies, what really was a driver for investing big time into these types of models was, in our case, a clear shift in our portfolio away from classic small molecules to complex molecules, large molecules, and oftentimes highly engineered molecules that that often also have multiple targets. So you can see that in this little graph there on the top right. So these are all bispecific antibodies, and for those people not familiar with that area, I mean, you can see, I mean, some of these molecules, they don't even look like antibodies. Oftentimes, they do not cross-react with any preclinical species, not even primates, which are typically the species of choice to assess large molecules. The targets and the pathways we're interested in are typically not adequately represented in any animal species, and here we're talking about everything that is immune-related. So, as I understand, this group here is particularly interested in these types of questions, so I'm sure you're well aware of the significant differences between preclinical species and human regarding the immune system, and that's also something that has been recognized by regulators here, exemplified by a key publication from the FDA some years ago. But this is not all. There's also additional upcoming challenges. There's an increasing need for assessing, for example, the safety and efficacy in children in different ethnicities. So these are questions we're getting from regulators. Sometimes we're dealing with a very small patient population, if you think of rare diseases. And there's the overall goal and aspiration to increase the benefit-risk ratio at an individual level, so strive for more personalized medicine. So these have really been drivers in our case, realizing that conventional preclinical in vivo testing may not be relevant or even may not be possible. So in the case of our large molecule portfolio, which mainly consists of, or to a good extent, consists of so-called T-cell bispecifics, there was simply no choice to go into animal testing. So we realized there's an urgent need to invest into these tools to assess both the pharmacology and the toxicology of these new types of drug modalities. I'm showing you this here. Some of you may be well aware of cancer immunotherapy is, of course, an important area where these types of molecules are being developed for. I still recall some years ago when in an internal meeting we proposed human cell-based models for the safety assessment of large molecules, that there was somebody in the audience saying, why do you want to do that? Large molecules, they typically don't have significant safety issues. And that was before the time drugs like these classic anti-CTLA-4s or anti-PD-1s were on the market. And I think in the past years, we all have learned that this is actually not the case. So exemplified by ipilimumab and nivolumab, there's a number of side effects documented in clinical trials that escaped preclinical safety testing. So this is the challenge we're all dealing with. So we went out to establish suites of human cell-based models to address what's called on-target off-tumor safety liabilities of these, in our case, bispecific antibodies. Most of these cell models, they follow the scheme outlined here. So based on primary human tissue, polarized where required, we try to introduce PBMCs if necessary and if possible in flow, and all those tissue and cell components, in the best case, donor specific, and if this is not possible, then at least HLA matched. So saying that, it's clear that current conventional 2D models, they lack essential cellular, biochemical, and biophysical factors found in the native organs that we're trying to recapitulate here using this novel approach. So the goal for us is to create models that are able to recreate first drug-induced activation of immune cells, then the initiation of downstream events, including cell migration to the site of action. And at the site of action, we would like to see measurable effects that allow drawing conclusions on drug molecule properties, both desired, so the efficacy, but also undesired effects, and to see how relevant is that to our current patient population. So to do that, a number of technical aspects need to be defined, and there's different types of assays and cell types that come into play here. So for the immune cell activation, you see here an example of the recruitment of blood leukocytes on the flow following TNF-alpha simulation. So this is an endothelial model here that has been developed in one of our labs. Then the targeting of healthy tissue, as we're focusing on safety in my former department. So here you see the infiltration into, in this case, hepatic spheroids following anti-PD-1 treatment. And then the effect, is there an effect that we should be looking at? In this case, it's the destruction. So we have a chemotherapy-induced apoptosis of intestinal organoids. So we really spent a lot of time with trying out and exploring and assessing and establishing and refining these models, playing around with different cell types, assessing what might be the relevant readouts and what can be used as positive and negative controls to actually validate those models for a very defined context of use. This is, of course, not all. This will not cover all the questions in this area. So what I'm talking about is mainly, as said, on-target off-tumor toxicities, which is typically restricted to tissues where the target is expressed. It sometimes may be less of a concern if already by target assessment, and we see that the safety window would be large enough, and if the expression in the tumor is very, very high. There's other types of questions that need to be addressed, which I will not talk about today, but just to make sure that to mention that these are, of course, in parallel activities which other labs and other groups are dealing with, and in the end should lead to a cell-based, human cell-based safety assessment. To name one of the challenges that we have been facing, and which some of you are maybe aware of, is if you want to include aspects of the immune system by adding PBMCs, is for example significant donor variability. So it's relatively easy to obtain PBMCs. You can match them, you can characterize them, you can choose the right HLA type. Nevertheless, there's a number of underlying factors which are driving significant variation, and that's something that needs to be carefully assessed. So this can really introduce next to a complex setting these sometimes flow-based organ chip type of cellular models anyway have. This is a significant additional source of variation that comes into the assay and makes it then oftentimes difficult to interpret. So this is something where we invested a lot of time actually, and still do. It's very model specific, it's cell type specific, and deserves always careful assessment how you deal with, for example, PBMCs in your system. Now, in the next couple of minutes, I would like to take you through a tour of some of the attempts. This is actually the first one, or one of the first one we dealt with, and there will be others following afterwards. So we were dealing with a molecule that shared similarities to a well-known molecule that was introduced a couple of years ago, urelumab, a CD137 antibody, which then showed quite unexpectedly at that time hepatotoxicity in clinical testing, and was removed. And so we tried to take this case, as there has been already some literature out there, as shown on the left-hand side. So this is a publication from some years ago where they used a mouse surrogate to see whether the effect of hepatotoxicity can be recapitulated. So we wanted to see, is there a way that we can build such a system that is able to recapitulate urelumab's infiltration of T cells into a mouse liver, and followed by toxicity? Can we build a system like that? So what we did then, so what was our plan and our vision, is to have a system like in this sketch on the left-hand side. So this is a 2-organ type of system where the idea was to have both the tumor and the healthy target organ of toxicity represented, in this case the liver, plus blood cells, which would be flowing through the system. And I hope you can see on the right-hand side, so this is the prototype that we developed together with the ETH in Zurich, the bioengineering laboratory. You can see this dark dot there. This is a liver spheroid, and you see the migrating immune cells through the system, which may look like a simple thing, but only to get there was actually quite a long way, and required a lot of prototyping with the system. So in the first attempts, we lost the spheroid because the gaps there were too big, then the flow was too strong, and so the spheroid just dissolved. If you reduce the flow, then the immune cells, they wouldn't move anymore. So there was a lot of work to be done in order to get to a point where I thought, okay, now we can give it a try and see if we apply our molecule from the portfolio candidate molecule can we recapitulate the T cell infiltration and subsequent toxicity. And as you can see here on this slide here, this is a summary of what we found. We were quite happy to see that that first of all we did see hepatotoxicity. This is in the first line. On the top line, you see the hepatocytes marked there with keratin 18 and the cell nuclei. And you can see with increasing amount of bispecific antibody, we can see hepatotoxicity here. And what was even nicer to see here is actually that this hepatotoxicity is due to infiltration of immune cells, which did not take place if we would use a non-targeted antibody at same concentration. So that was a very, very first data that we generated about, I would say 5 to 6 years ago, and very encouraging. It was difficult to get there, but this was for us a game changer and a proof that yes, you can recapitulate such complex biological processes in an in vitro setting. So a number of new and additional approaches then were kicked off. One of them I would like to show you is that after some years, it could finally be published last year. So this stems from a collaboration, from a multi-year collaboration with the Boston-based startup Emulate. So what we wanted to do is make use of their existing lung on a chip model that you can see in the middle here. I'm sure most of you have seen this model where you have a top channel lined up with human epithelial cells. There's the vacuum channels on the left and the right that we didn't use for our purposes. The epithelial cells would sit on a porous membrane, and below there, representing the blood vasculature, are endothelial cells, where we would apply PBMCs under flow. So this system we wanted to use for a case from our preclinical safety in vivo group, which used a TCB that would target CD3, so for the T cell arm, and folate-1 receptor as the tumor target. So the model, the molecule has been shown to be efficacious in human breast cancer patient-derived xenograft models, and then was moved into a first PK study in cynomolgus monkey. It was known based on the target assessment that the target is expressed also on the apical site of lung, of both human lung and also of cynomolgus lung. But it was believed at the time, because there is still this epithelial barrier, chances are low that the T cell bispecific would actually reach the target there. And the team unfortunately had to learn that this is not the case, because in that PK study, they observed leukocytic infiltrates in the lung tissue indicative of immune-mediated toxicity. So in order to prevent further cynomolgus in vivo studies, the team has asked us, could we use this refined Emulate chip to test candidates before moving forward into in vivo testing. So we looked into the chip, and as you can see, we could confirm actually expression of folate-1 receptor, confirming that the system could be relevant for this type of effect. So we then did a number of different experiments to see whether we can recapitulate the lung toxicity. I'm showing you only a few of them. So what you can see here in this graph are apoptotic cells collected on live chips, so shown by caspase 3 positive cells. So what you can see here, that there's only caspase-3 positive cells if both PBMCs are present in the system and also the folate-1 receptor targeting antibody. So there was no effect with a non-targeting antibody, and of course also no effect if there were no PBMCs present. So that was already quite encouraging that this model seemed to be working. We did a number of additional assays. For example, as shown here, we could observe PBMC accumulation at sites of target expression. So when you look where the target is expressed, you can see that PBMCs seem to accumulate primarily where there's a CD3 positive cells around the target, which is also then quantified on the right-hand graph. So this is the total of CD3 positive PBMCs that could be observed, where again, only when the targeting antibody was used. We did another round then of experiments trying out different versions, newer versions of the same antibody, a low binder and a high binder, to see whether we could identify a molecule that would still be efficacious, but which would have a more beneficial safety window. And as you can see with the low binder T-cell bispecific, the toxicity only is apparent at higher doses. So that was kind of a very encouraging first attempt to use an organ chip model to identify a safer molecule that could then later on be moved into in vivo testing again. Moving on to another example where we used a different platform. This time it was more about the basic question whether we could recapitulate neutrophilic infiltration into an intestinal model of inflammation. So to support questions around inflammatory bowel disease and other types of autoimmune dependent inflammatory diseases, and see whether such a model could be used to support testing different drug candidates and refining them and choose the best one to move forward. So the system we used for that is the Mimetas system. Oops, that was sorry. Which is like you see on the left-hand side. So it has the same size as a 96 well plates. You have 40 microfluidic chambers, which have 3 lanes. We used 1 lane to grow an intestinal tube. Then in the middle, an extracellular matrix, and the right-hand channel we used to apply medium and fluid. So we could generate a polarized intestinal epithelium, and actually, as a first assay, develop a test to assess drug-induced leakiness of the gut, which is being used now. There's also other types of approaches also published, which address the same questions. We wanted to see whether we can use this system to see whether we can mimic the aggravation of intestinal inflammation if we have both resident and infiltrating immune cells. And this is what's summarized here. So when you go to the left-hand side and you have kind of the normal situation with the healthy gut, then with the infiltration of immune cells, the gut gets leaky. It aggravates. You have the activation of macrophages, which then leads to neutrophil recruitment, and then the whole system entering in some sort of a vicious circle where the situation starts to aggravate more and more and more. So this was the system that we developed here, the system for intestinal inflammation. So this is a triculture of epithelial, resident, and infiltrating immune cells, which allows to capture their functions and functional interactions. So far so good. That was really the first attempt. It's still very basic. We could show that infiltrating neutrophils exacerbate the inflammatory process, leading then ultimately to epithelial damage. This is not the perfect system. So at that time, the first system was still Keiko-based, so cell line based. This cell in such a setting actually displays quite nice improved features over normal culture methods in 2D, but still it's not primary cells, so newer versions of the same system contain primary cells. And of course, we're missing stromal and vascular cells, and not forgetting also what is important for this type of disease setting, also the adaptive immune cell contribution to the inflammation. So this is all not captured. It's still a very simple reductionist system, but that was at the time we developed it, again, was quite encouraging for us to see that it is possible to mimic such a situation in an organ chip type of model. So switching gears now, this is an example where we contributed to candidate molecule identification for our oncology team. In this case, it was about 4-1BB agonists for a combination therapy with T-cell bispecific antibodies. The system that was used there was again a totally different one. It looks quite funny when you look there on the left-hand side. So it has this U shape. This is this U. It's called U cup. It's stable for only a limited period of time. So it has some drawbacks and it's a bit complicated to handle, but for this type of approach, it was used and it could produce actually quite nice results. So you have there in the middle of that U cup, you have this collagen scaffold, and in between tissue blocks with patient-derived tissue. And you can perfuse it then with medium and PBMCs. So this system was then used to see whether a combination of TCB with tumor-targeted 4-1BBL agonists could help improve drug effectiveness. Could we recapitulate functional cross-linking of TCBs with FAP on human tumor tissues? And how would that relate to what is observed in vivo? So you can see on the left-hand side, so this is the target expression in the tumor tissue samples, and you can see on the right-hand side, these were the experiments that have been performed using the UCUP system, and you can see nicely when adding the cross-linker that you have a significant improvement of a total amount, an increase in total amount of interferon gamma, indicative of an improved effect. So that was again confirming that such a system might be very useful to test different type of approaches and also different types of molecules in a real patient tumor setting. Now, switching gears a bit, this is something which is not related to directly to immune-related questions. I just built that in from another presentation, just because. Yeah, I like the example. You know, it doesn't always have to be super complicated. So this is an established system that's around for some time. It's an OECD qualified test for eye irritation. And we wanted to take the system to a higher level to address a different type of question. So this is the 3D corneal epithelium model, which serves as an in vitro model for front of the eye applications and eye irritation test. So the test is outlined at the bottom, so it's relatively simple. You have a transwell, you have medium at the bottom, and you can add drug from the topical side, and then test viability. So this is the established test. And the team now wanted to use this test to see, can we turn this into what we would call a dry eye disease model? Certainly not the perfect one, but one that is relatively easy to get to by simply changing the culture conditions. And as you can see here in the picture there, in the image on the left bottom, you can see the dry eye phenotype actually quite nicely. So it leads to a decrease in the epithelial thickness after just 48 hours. And this was then used again to test different types of formulation, which have, of course, a strong impact then on the efficacy of the drug, and also would significantly reduce the use of animals, which in this case would have been cynomolgus monkeys. So we just like this example also to show that it doesn't always have to be something super complex, can also be just an existing model, tweak it a bit and adapt it, and that can already really support efforts that otherwise would have taken quite a lot of animal testing, or at least a larger study. Another example that I would like to show you here is an organoid model. So we invested also quite a bit into organoids, and I come to that also a little bit later on. There's some newer developments. This one here I like to show because probably you wouldn't think of a blood-brain barrier model to be the best approach for an organoid. But in this case, actually, it worked quite nicely. We could culture the organoids in a newly developed system by Sun Biosciences, which allows to culture up to 3,000 organoids per plate in a highly reproducible size and compatible with automated microscopy. We observed that when mixing together different cell types, so in this case endothelial cells, astrocytes, pericytes, that they would self-assemble into what can be considered some sort of a neurovascular unit. And then by using molecules, in our case, we call them the brain shuttle. So a molecule that should help a shuttle small molecules, transport them into the brain. So they could be tested using such a system. And this is what you can see here. You can see on the left top side how our brain shuttle is built. So you have the antibody payload and then transferrin receptor binder. And we produced a number of different variants. There was the lead molecule, there we had already significant in vivo and also clinical data. So we could use this to validate the whole system, and then a number of follow-up molecules to see whether we can improve the properties of that brain shuttle to identify better molecules that would even be show more, in this case, transcytosis and would transport more drug molecule into the brain. So just as an example of organoids as a relatively simple system that can really accelerate optimization of current complex molecules. Now, where do we want to go? What is our current interest? So if you think back of the example where we used our cell-based models for T-cell bispecifics, so we do have the ambition to actually base the safety assessment on in vitro only, so both the efficacy and the safety for entry into human enabling all based on in vitro system. So what can be considered an in vitro therapeutic index. So you have the tumor where you of course expect a strong effect, and then test a number of additional healthy organs, so including those that have been identified by target assessment where the target is expressed as well, plus adding also what we call essential vital organs. So just to have a bit of more insights on different types of cells. So we are running this approach currently. It's quite a challenge, but we are quite positive that this is something that will allow us to move molecules without any safety in vivo testing into first-in-man trials. So this is one of the focus areas for us. Another area of focus is make direct use of patient-derived cells to see whether these patient-derived models can help us personalizing clinical trials. So there's, again, this comes with lots of opportunities, but also lots of challenges, starting with access to those tissues, to informed consent, and also defining a path and an algorithm where these types of models, which oftentimes require a lot of time until they can be used for testing. So to have them in really a very short time from patient, from deriving from the patient the cell to a ready, to a assay ready format, and then taking decision based on the in vitro assay, so that this all fits into a clinical setting. So this is a newer area I'm focusing on with the team in clinical safety, but we already have been able to generate some quite promising first data. Now, going back to the organoids and these type of newer models in general. So Roche has recently founded a Roche-owned internal institute for translational bioengineering that entirely focuses on organoid-based approaches to support both our discovery but also our development organization. Matthias Lütolf, who is certainly one of the pioneers, is the director of this institute, and together with his team is focusing on developing robust assays, fully automated assays to serve our disease areas. So I think this is a nice signal and also a strong commitment of the company to really build on these novel type of approaches to support drug development. Now, summarizing up some of the approaches and some of the ideas and visions I alluded to have been part of mainly two publications that I was happy to be able to release last year together with colleagues about where we would like to go, where we see this field evolving. So we have seen significant investments in both academia, biotech, and pharma, which have led to a series of encouraging use cases that demonstrate the potential of these human relevant cell models. We have also seen that the adoption in industry is relatively slow for various reasons I alluded to, so there's still quite some work to be done. I think there's areas of high unmet need, which could really be an opportunity, and these areas could really help driving the application forward. So where the bar for adoption and application is lower than in other areas. So I hope I could convince you with the example around cancer immunotherapy, where some years ago we were really confronted with basically a lack of good tools. and the organization was ready to accept and is currently ready to accept that some of these systems are not perfect, yet it's still better to use something that is still exploratory in nature but can support drug development at these stages, and the models can be refined with every new program that is coming up. I also see opportunities for these types of systems where in areas where lean drug development paths are possible, for example, rare diseases, where also regulators have different expectations regarding the maturity and of the types of tools that are being used. And also, I think we've seen now during the pandemic, there have been a number of examples where these tools have been used. If you think of, for example, Harvard's long on a chip model for repurposing approaches for COVID. So testing marketed drugs and see whether you can identify one that might be of use to fight the pandemic. So I think these are some of the areas where I think these models could be adopted much faster than in areas where there's lots of established tools already around. I think, of course, now also thanks to my new role, these models can not only significantly improve our preclinical development phases, but could as well become also game changers in clinical development. So personalized medicine is one of the areas where we currently look into. And just to say it again, so next to access to patient tissue and cells, which I think is really absolutely key, especially if we think of immune-related aspects, this is really a challenge. Also, the access to patient data is important and is not always easy to get. Ideally, of course, from both from the same patient, both the tissue and the data. So this is a challenge and is something we're currently working on. So I hope that was interesting. With that, I'm already at the end, and I would like to thank, if the slide is moving forward.